So just wanted to highlight all these notes on my whiteboard, which my daughter writes, and then I have to work around it because I never wipe them off because I want to keep it as much as it is. So I really don't have much space to work around my whiteboard anymore. I'm Saurabh Kulkami, and I'm an assistant professor at UVA, and we are trying to figure out how to do rapid functional genomics of variants identified in heterotaxy and PCD. So this is the image of multiciliated cells of the frog embryo, and I'll show you a little bit more about how frogs are great model systems to actually address this question. So I just wanted to highlight some of the challenges in the genetics of heterotaxy and PCD. And while we know that basically because of long read sequencing and whole exome and whole genome sequencing, we are actually sequencing patients a lot. But translating these genetic discoveries into actual clinical diagnosis has been really a biggest barriers in the genetics. And there are multiple reasons for it. For example, there is a huge genetic and phenotypic heterogeneity in heterotaxy and PCD both. Also, basically sequencing gets you the variants, but how to actually interpret it is actually lagging behind significantly. So interpreting missense variants have been actually one of the biggest problems in the genetics of these diseases. Also, we can use in silico models now. There are tons of them, but they have a really limited accuracy, so you can't really use them as efficiently or as reliably. And so they only give a PP3 level, a probabilistic level of pathogenicity for you to actually interpret the VUS that was identified. And also, some of the VUSs are context-specific, like some VUSs may just cause heterotaxy, but not PCD or neurodevelopmental disorders, and we still don't understand that. And in silico models cannot actually tell us what VUSs cause what kind of phenotype. We also do not have in vivo model systems or actually disease-relevant model systems that we can use to study these diseases. And finally, we don't have a clinically integrated functional genomics pipeline, which we need for PS3 level VUS classification. So I'm just going to give you an example of one of the patients that we recruited at UVA. I'm not a clinician, so do not ask me anything about the patient phenotype. I'm just giving it to you, saying that he had a heterotaxy. But I talked with the clinicians. They said that he doesn't have any respiratory complications identified over the last 2 years. There was no sign of PCD in the patient, and basically no extracardiac malformations. So we did whole genome sequencing for the patient, and we identified 2 mutations in DAW1. One was a compound het mutation, one was a missense mutation, R114Q, and the other one was a splice site mutation, which leads to donor loss. So first thing we did was to just look at the in silico predictions of these variants. And as you can see, for the missense mutation, the variant interpretation ranged from completely benign to completely pathogenic, rendering it as a VUS. So this it depends on what in silico prediction tool you're using. You can get any result from benign to pathogenic, and this is really common for missense mutations. And for splice site mutation, there can be loss of function, and so this time the in silico prediction was much better. They were all deleterious, so we knew that because it's a donor loss, it's keeping the entire exon 7, and it might be pathogenic. And so just to give you a little bit of a DAW1 biology, so DAW1 is a dynein assembly factor with WDR repeats. So it actually is important for motility of cilia in the left-right organizer, or the node that we heard so much about, and also in the multiciliated cells of the airway. And there was a study, or 2 studies published previously, identifying DAW1 mutations in patients with heterotaxy, with respiratory or PCD-like symptoms, or without PCD-like symptoms at all. But we really do not understand why some mutations give heterotaxy and why some mutations only give PCD. And so there are 2 questions we are trying to answer here. One is, are these DAW1 variants responsible for the patient heterotaxy? because both of them were VUSS. And the second thing is, can we explain why the patient has heterotaxy but not PCD? Because DAW1 is essential for motility of cilia in both airway multiciliated cells and in the node. And so here I'm going to show you why frogs are cool to actually understand this, because you have learned about mouse before, and a lot of studies on the mouse have been done. People have used zebrafish, but I can bet you they cannot do what frogs does, and you can see it here. Can they do that? No. Frogs cannot do what mice, frogs can do what mice and fish cannot do, okay? So for the science purposes, they are cool for science too, right? Because frog eggs develop outside, unlike mouse, right? You can actually study every developmental process of the frog under the microscope, right? So this is where the frog is just going gastrulation and forming the left-right node. This is actually an axolotl embryo, but very similar. Now the embryo is forming, and you can actually look at the left-right patterning during these developmental stages using molecular markers. And now the axolotl embryo, which also looks very much similar to frog embryo, you can actually look at the heart itself. So I really don't have time to go slowly, but you can actually study these embryos outside and actually look at every developmental phenotype that we are interested in looking at. So we generally, what we do is we get like about 3,000 to 4,000 fertilized eggs every time we do the fertilization. And we can inject, if you want to inject a human mRNA in there, so human gene, or we can knock out or knock down a protein and look at the heart of the frogs. Here, one thing I want to basically highlight is basically this cilia in the node are monociliated. So every cell only has one cilia, and they're fundamentally different from the airway multiciliated cell. And so we look at the organs in the frogs. For example, here I'm looking at the heart looping or cardiac looping or outflow tract. And actually I can tell if this frog is actually developing heterotaxy or not. And I'll go a little bit more detail in that. And you can also look at the other organs like gallbladder and intestine and see if they have switched their places. We can also look at the multiciliated cells. So a lot of these heterotaxy patients develop PCD, but studying these multiciliated cells in vivo in mice is really hard because they are inside the airway tract, right? So what should we do about it? Because we have to kill mice to actually look at their function. But in frogs, the entire embryo, which is, this is 24 hours post-fertilization, the entire embryo is covered with multiciliated cells. So all these green dots you see are multiciliated cells that function just like our airway multiciliated cells. Okay? So I'll show you a little bit of video. So as you zoom in on this frog, you can see all of these multiciliated cells. They create flow just like our airway multiciliated cell to create a flow from anterior side of the embryo to the posterior side. So here is a high resolution image of a single multiciliated cell. You can also look at all the basal bodies and cytoskeletal structures inside the cell. And you can also visualize these multiciliated cells beating on a live embryo without killing an embryo. So you are looking at its development in vivo. And you can also do the bead transport assay, what we talked about yesterday. Basically, you put the red beads in front of the embryo, which is the head of the embryo right there. And these beads are actually directed from the head to the tail using these multiciliated cells beating on the surface of the embryo, right? So these beads would be exactly the same way be actually transported in our airway to clear them out of the airway. So coming back to these questions, can we actually use frog as a model system to address these questions? So first thing we did is basically we took these frog embryos and we inject them with the morpholino to deplete DAW1 or take it out of the frog. And then we saw the heart development, gallbladder and intestine position, and you can see that this is the normal position of the heart looping or outflow tract. But when you have a heterotaxy, you can either get a heart that is not looped at all and the gallbladder right in the center, or you get heart that is looped other way around and the gallbladder on the right side of the embryo. And when we do that, we find that when we knock out DAW1, we get about 60% of the embryos which have heterotaxy kind of phenotype. We can also use the same exact embryo and look at the flow or mucociliary flow and multiciliated flow in these embryos. And when we do that and we put the red beads in front of the control embryos, you can see the beads are actually flowing nicely in front of the embryo. But when we remove DAW1 from the frogs or deplete it, you can see the beads stay in the same place and they do not flow at all. So these are not different embryos. It's the same embryo looking at the multiciliated cells and at the heart looping. And we can show that basically embryos in control, they have all flow. In the DAW1 knockdown, they don't have, or 90% of the embryos do not have any flow, showing that DAW1 is essential for motility of the cilia and mucociliary clearance possibly. And so the question now is, where is DAW1 localized, right? And so this is the localization of DAW1 in the center. So this is a single multiciliated cell from the frog embryo. And this is the cilia or axonyms labeled with the membrane. And this is the DAW1 GFP of human DAW1 GFP, not Xenopus, right? Localized in the axonym of the Xenopus cilia. And this is the overlap. And so now we are going to use this to actually study the mutants. So this is, instead of now generating a human wild type DAW1, we also generated a patient missense mutated DAW1, okay? And we injected it and looked at the localization. So the missense mutation didn't really change the localization of the protein from the axonyms of this cilia of multiciliated cells. But here I'm going to walk you through this image graph. So here you're looking at a heart looping or cardiac looping graph. On the x-axis is your percent embryos, and you can see all control embryos had normal heart looping or outflow tract. When you deplete DAW1, you get about 60 to 65% of the embryos who have heterotaxy like a phenotype. Now, when you inject human wild type RNA to replace the frog DAW1, you are getting almost 80% rescue of that phenotype, suggesting that the human DAW1 works fine in Xenopus, and it can be used to test the patient mutations. And now, instead of injecting wild-type RNA, we are injecting human patient mutation and show that basically we cannot rescue the cardiac looping phenotype, suggesting that basically the patient mutation is pathogenic or it's not functional, even though it localizes properly. Now, we are going to take the same embryos and look at the flow of the multiciliated cells. And so here is the mucociliary flow. Here again, all control embryos had complete flow. When you knock out DAW1, you get 85% of the embryos that do not have flow. When you inject the wild-type RNA of the human's DAW1, you get a complete rescue of flow, almost 80 to 85%. And when you inject the missense mutation, which is not functional in cardiac looping, in the same embryos, can still rescue the flow of multiciliated cells, suggesting that mutation is pathogenic in left-right organizer cilia, but it's not pathogenic in the multiciliated cells of the airway or multiciliated cells of the Xenopus, suggesting that maybe patient is not having a PCD because this mutation is not pathogenic in the multiciliated cells of the airway. Now we do the splice site variant. The splice site variant is predicted to be pathogenic, and it is keeping this exon 7. So when we do AlphaFold modeling, you can't really see any difference in structure of the protein. But the exon 7 here is skipped from the DAW1. When we look at the localization, it's a complete loss of localization. So DAW1 does not localize to the cilia or the basal bodies at all in these multiciliated cells, suggesting that it's a complete loss of function. And so we did the same rescue with the splice site for heart looping, and we do not rescue the phenotype at all, suggesting that the splice site mutation is a pathogenic mutation. And we also cannot rescue the mucociliary flow or multiciliated cells, suggesting that splice site mutation also affects the mucociliary flow of the Xenopus embryos. But we have to remember that basically because the autosomal recessive nature of the PCD, since R1142 is not pathogenic, you can still maintain the mucociliary flow. However, in the left-right organizer, because both mutations are pathogenic, you are getting the left-right patterning problem. And so, just to conclude, we already established that frogs are way cooler. They can be used for CHD heterotaxy phenotype. They can be also used for the PCD analysis of VUSS in the same animal system. And the thing that I have not shown is basically they can also be used for neurodevelopmental phenotype by looking at their brain because they are transparent. The second thing is in frogs, we can actually generate genotype-phenotype correlation and show context-dependent functional assessment of the variants, why they are functioning to be why they are functioning to be pathogenic in one context but not other. Third thing I wanted to emphasize that is why I think we need a disease-relevant cell type or a tissue type to study the function of variants, because if we are studying them outside of the context of disease, we may not actually interpret the pathogenicity or benignness of the variants appropriately. Also, loss of functions are function variants are easy because they're loss of function, like truncating mutations. But missense mutations are really hard because they can be gain of function, they can be hypomorph, they can be loss of function, or benign completely, or even dominant negative sometimes. And so basically, to address these challenges, in my lab, we are trying to actually develop a rapid functional genomics pipeline, because in this case, we actually kind of figured out the pathogenicity of these mutations in less than 2 months after we received the genetic diagnosis. With that, I want to acknowledge the lab tech in my lab who did all the work. The rest of my lab, the Halloween competition that we win every year in the department. The clinicians, Christina, Matt, who's the genetic counselor, Andrea, you probably all of you know, who runs PCD clinic at UVA, and Akrosh, who does bioinformatic analysis of whole exome and whole genome sequencing, all my collaborators in funding. Thank you.